Welcome to the International Institute of Technology and Applications for Clinical Research, Drug Development, and Devices. In today's lessons, we will discuss the phases of clinical trial research. It includes the introduction of a compound or device to the human population. It's done in a series of steps. In this slide, we will focus on the phases of clinical trials. The preclinical phase focuses on lab and animal studies. Phase 1 involves a small number of volunteers with a focus on safety studies. Phase 2 consists of hundreds of volunteers where we identify the proper doses of drug for safety and efficacy. Phase 3 consists of thousands of volunteers in order to confirm the efficacy of the given drug. In Phase 4, we monitor the general population for continued safety and any detection of long-term adverse effects. Phase 0, typically known as proof of concept. Normally this isn't covered in regulations, but is being used more to minimize resources in moving to Phase 1 with a compound that isn't going to pan out. An emerging technology is microdosing, where a minute amount of compound is introduced into the human subject. This allows for the assessment of pharmacological and mechanisms of action without significant risks of toxicity. This phase is normally used on healthy volunteers, except in oncology. Phase 0 consists of testing for pharmacokinetics at the subpharmacological levels, better known as microdosing. This phase also considers single rising doses to identify levels of toxicity. In this phase, we also plot the plasma concentration against time, where we look at the area under the curve to determine levels of toxicity. After drug administration, we estimate the bioavailability and clearance of drugs. The primary goal of Phase 0 is to identify the pharmacodynamic and pharmacokinetic properties of a drug, particularly oral bioavailability and half-life of the drug. Typical doses consist of 1/100th of a therapeutic dose. Phase 1. In this phase, researchers test an experimental drug and treatment in a small group of people for the first time, basically to evaluate its safety, determine a safe dosage range, and identify side effects. The initial group of people are usually healthy volunteers or subjects, except in oncology. These trials are often conducted in an inpatient clinic where the subject can be observed by full-time staff on a 24/7 basis. Phase 1 continues with the investigational agent tested for the first time in humans, designed to evaluate safety, not the efficacy claims of the agent. This is typically done in normal, healthy volunteers unless toxicity contraindicates. Designed to determine metabolism and pharmacological actions of a drug in humans, as well as also determining the side effects associated with increasing doses. At what dose? Usually a lab range or upper lab value is predetermined, and the dosing is stopped if and when that point is reached. Phase 1 facilities. This is usually staffed 24/7 with medical personnel on site at all times, using inpatient population and controlling activities, food, medicine, and so on. Phase 1 continued. Pharmacokinetics and pharmacological effects should be obtained to permit well-controlled, scientifically valid Phase 2 studies. Pharmacokinetic studies consist of absorption, distribution, metabolism, and elimination. What the body does to a specific drug after administration. Performing serial blood draws based on the time of dosing. Therefore, one mistaken sample taken at the wrong time can throw off the analysis significantly. Pharmacodynamic studies. This is what a drug does to the body. In that, we take physical assessments such as lab work, continuous heart monitoring, and physical exams. Activities for phase 1. This is also called a pharmacological phase. That consists of single ascending dose, special studies, genotoxicity, carcinotoxicity, repeated dose, reproductive toxicity, tolerance, safety, and pharmacokinetics. Phase 2, A and B. In phase 2A, we determine how much drugs should be given. Phase 2B, we determine how well the drug response is to the dose. In phase 2 trials, the experimental treatment investigational product is given to a larger group of people, usually between 100 and 300, and this is to see if it is effective and to further evaluate its safety. Phase 2A. The primary goal is to further determine safety. The secondary goal is to determine the therapeutic efficacy. Pilot trials. Dose response determination, determination dose regimen, determination of target population. Subjects, a few hundred. These subjects will fit a narrow eligibility criteria. Trial design, comparison with baseline status, open label, no blinding, or single double blinding. Dose escalation, to determine the minimum tolerable dose. Parallel dose response, choice of control groups. This includes placebo or comparative marketed drug against the investigative drug. Phase 2B. The objectives here is to focus on aspects of phase 2A trials. This includes blinding, placebo or other concurrent control, crossover treatments, multicenter. Trial design. This could be single or double blinded, and it's conducted at multiple sites. Phase 2, continued. Clinical trials are also part of the early development stage and primarily focus on drug safety, but also starts to collect data on efficacy. Data is collected to determine and confirm safety, efficacy, clinical endpoints, dose, toxicity, short-term adverse events. Phase 2 also provides the foundation for required Phase 3 trials by providing clinical endpoints and determining potential dose levels. The main goal of Phase 2 trials is to determine whether there is a therapeutic justification and a commercial rationale to move into Phase 3. Safety, primary endpoint, and effectiveness is a secondary endpoint. Relatively small numbers, generally in the low hundreds. Phase 2 trials is used to a selected population of subjects who have the disease or condition to be treated, diagnosed, or prevented, typically earlier onset or less severe diagnosis with minimal comorbidities. Phase 2 trials are conducted with control arms, although placebo control arms are not always used. For example, in cancer studies, the use of comparative market product is recommended for the control group in oncology cancer. More diversity in design studies. This phase of the trials are conducted to determine minimum effective dose. Safety profile is examined in relationship to dose. What adverse effects are more commonly experienced at a particular dose. Phase 2 trials are randomized, double-blind studies designed using a placebo group and a low, middle, high investigative product dose groups. They are multicenter trials. The monitor in this case will not be present at the dosing of subjects in most cases. The CRA must facilitate compliance by ensuring adequate training. Activities for Phase 2. This is also known as the exploratory phase. This consists of product interaction, route for administration, that is either in the form of a tablet or intravenous, efficacy, safety, schedule. Dose response and the trials must be controlled, randomized, and adequate monitoring for accuracy, completeness, and verification of data. Phase 3, A and B. Phase 3A, subject would be large. Trial design, open label or single or double blinded. It's multi-center. Testing, different stages of the disease indication. Dosage, forms and formulation. Different routes. Pharmacoeconomic evaluation. Phase 3B. Phase 3B trials will continue while regulatory submission is pending at the appropriate regulatory agency. This allows patients to continue to receive possible life-saving drugs until the drug can be obtained by purchase. In Phase 3 trials, the treatment is given to large groups of people, 1,000 to 3,000 or more, to confirm its effectiveness, monitor side effects, compare it to commonly used treatments, and to collect information that will allow it to be used safely. Subjects always have a pre-existing condition. Phase 3 continued. Many considered as the pivotal studies in which marketing application is based. Large, multi-center, well-controlled investigations involving several hundred to several thousand subjects. The number of subjects is determined by agency through meetings and/or statistical considerations. This is the most expensive phase. Efficacy. Phase 3 looks like drug interactions, expanded safety profile, and extended use of drugs. Phase 3 trials, the designs can be randomized, double-blind, comparator, crossover. This phase evaluates utility in targeted diseases. Usually, primary endpoint is efficacy and secondary is safety. Activities for Phase 3. This is also called a confirmative phase. This includes product stability, efficacy, target population, safety, final formulation, storage, benefit, risk, analysis, and it's also controlled, randomized, and adequate monitoring for accuracy, completeness, and verification of data. Phase 4. This is post-marketing surveillance. Phase 4 trials involve safety surveillance and ongoing technical support of the drug after it receives permission to be sold. The safety surveillance is designed to detect any rare or long-term adverse effects over a much larger patient population and longer time period than was possible during phases 1 through 3 clinical trials. Harmful effects discovered by Phase 4 trials may result in the drug being no longer sold or restricted to certain uses. Phase 4 continued. The goals: to gain additional information on the drug, to revise the labeling and package inserts, long-term safety and effectiveness, to support claims and enhance advertising. How much does it impact the day-to-day lives of patients? Do doctors choose to prescribe it relative to other drugs available? And what about the market performance? Do new uses or indications emerge? As a result, this will usually require new application and more studies. Activities for Phase 4, also known as the therapeutic use phase. This consists of comparative effective studies, assessment of adverse events or reactions in larger populations, and the pharmacoeconomics of the drug or device, and marketability, market response. In our next lesson, we'll discuss the regulations and guidelines as it affect clinical research.